vaccinating, you introduce a small piece of the virus or a part of the bacterium or a heat-killed germ,
This is our
and the body sees it
coronavirus
and says, "Oh, this isn't normal. This doesn't belong here. Let's
virologist
attack
that
it.
is
Let's
going
make sure
to
this doesn't cause a
be
problem." And so
working
the
on a
body
team with several
puts
countries
together a response.
to get us the
It makes
COVID
proteins that fight infection. With the virus,
vaccination.
these proteins often neutralize the virus,
I'm taking what he's
and those
making.
proteins are called
Let's
antibodies.
listen.
They're made by a particular kind of immune cell called a B cell. At the same time, the body's making, we call them killer cells and helper cells, and those fall under the T cell category. So as the body's making these cells, the killer T cells are able to attack cells that have been infected by the virus, or the helper cells. Basically, they're kind of like the source of information and communication in the immune system. So your helper cells tell the B cells, make antibody. The helper cells help the killer cells by giving them the right kinds of signals to go ahead and mount a full attack against the virus.
So they're all part of like a white blood cell, is that?
They're all white blood cells, right? And the white blood cells defend you against infection. They're not the only white blood cells, because there are some white blood cells that aren't specific to particular pathogens, but they exist primarily to defend you generally against infection. They're like the fence on the outside of a camp, whereas inside you have the soldiers, the cells that are trained specifically to fight against this virus or that bacterium. And it's to the point where, you know, some of these cells will not react. So if you have a B cell that is specific for smallpox or COVID-19, and you come in with a completely different virus, a virus for rotavirus or respiratory syncytial virus, that B cell is going to say that nothing, won't react. It really is highly specific for the pathogen that induced it. And actually, that's a good thing, because you don't want your B cells, just cranking out antibodies
Just fighting everything. Yeah.
Right. Yeah. I mean, what you want is something that is really directed exactly at the pathogen. If we use the whole killed virus, it would be the entire virus. The outer coat, which has 3 or 4 proteins, the inner coat, which has usually a mixture of different proteins as well. Often with a vaccine, we can actually focus the body's defensive response on the 1 or 2 things that are the most important. And so, unlike a natural infection where your body's responding to everything in the virus, with a vaccine, we can actually tell the body, you don't need to respond to everything. Respond to this, and that will kill it. So in a way, the vaccine allows the body to be smart about what to attack.
And you can control that dosage?
Actually, you can control it by how much of the vaccine you give. In fact, one of the things you do when you're testing is you increase the dose to see what the best dose is that induces the best response. Sometimes you want the response to occur after the first dose. You may give slightly more. And we know this for several vaccines. You have to space out the doses in order to get the maximal response and a response that will last a long time. So when you're developing a vaccine, sometimes you're interested in a lot up front very quickly after the first dose. Sometimes you're waiting for the immune system to get to be appropriately reminded. So you give 1 dose, and then you give a booster dose, and then you give another dose. And that actually allows the defensive response to become better. The more times your body's immune system is exposed to a pathogen, to a germ, the better and faster and bigger the responses get. So that when a child gets 3 vaccines, every time they get a booster, the response goes higher and comes up faster and is much more specific. This is just the way the body has adapted to living in a world full of germs. We're lucky that we have this specific immune response. We call it the adaptive immune response.
Why does a vaccine take so long to develop?
So, right, under normal circumstances, with a standard pathogen that we're interested in developing a vaccine against, it could take 5 to 10 years. And that's pretty standard. If you look at the vaccines, sometimes they're 4, sometimes 6, and sometimes 10 or more. I mean, we've been working on an HIV vaccine without one.
So why do we not have HIV vaccine if so many smart people have been working on this for a decade?
That actually is the $1 million question. So, and this gets to why we think also that a COVID vaccine may be more possible. So remember I was talking about how after you've gotten an infection, you're protected. That's because, so say you develop COVID, you acquire COVID-19 from, somebody who's coughing. As your body is mounting that response, it's making the infection-fighting proteins called antibodies. It's developing the killer cells. And that combination of antibody and cells drives the virus out of the body, gets rid of it. The second part of it is that who's protecting the body? Why is the body immune to a second infection? Well, it's because these infection-fighting proteins and killer cells are there, and they're ready and they're waiting. And if they ever see COVID again, they immediately, significantly increase the number of antibodies and cells, and they keep the virus at bay. So knowing that makes it much more likely that we can develop a vaccine. The problem with HIV is that it doesn't behave that way. With HIV, you can be infected with HIV, but you can be infected again. So we know that you can be, we say, super infected. So if you are completely controlled on drugs, right? So with HIV, we can treat people and eliminate the virus to undetectable. If you happen to have sex with someone who has a resistant virus, you can acquire that resistant virus and it could take over. So super infection, as we call it, the lack of protective, durable immune responses in an HIV-infected person is one of the reasons it's so difficult to develop a vaccine. One of the T cells helps everything. It's like the controlling, the communications center for the immune system. HIV attacks that cell and kills it. That is the target cell for HIV. So now all of a sudden your immune system is without an effective way to communicate to the B cells and to the killer cells. The other thing is that the virus, the HIV virus itself, has involved a whole series of protective mechanisms. It mutates itself very, very frequently. So it changes the proteins on the surface. So in an HIV-infected person, you can have billions of slightly different viruses in your blood.
Wow.
I mean, my lab, when I was still in the Army, used to sequence these viruses. And, we could sequence 1,000 very easily. And they're all different.
COVID-19 is not like that.
So we think it's still one of the unanswered questions, but the belief is, and you see this manifested actually now in everyone's belief that if they expose themselves to COVID, they might become immune and therefore they can go to work. We think that COVID-19 infection is protective against future infection. Okay? So it gives you immunity against reinfection.
That's what we heard as well from Dr. Fauci as well as Professor Kim. So far, they had a lot of reactivated cases or relapses, but they haven't seen any case of reinfection.
Right.
Is that correct?
So the problem is that it's difficult to tell. One of them is, I think that the tests are not sensitive enough. So, you have you're recovering from the virus infection, and the amount of virus in your blood is decreasing, and the amount of virus in your throat, the places that they're swabbing, is decreasing. And finally, you reach a level of virus where the test can no longer pick it up, but it's still there.
We heard that that's around below 3,000 cells.
It's actually, I'm not sure that we know the count, especially on the throat,
Right.
where that level of sensitivity is. But with HIV, for instance, when we first started measuring the virus in the blood, the lowest we could go was 1,000 per cc of blood. And then it was 400, and now it's 50, and it's going below that soon. So if you were negative at 1,000, it didn't mean that you were negative. It just meant you were below the level of detection. So one of the potential explanations is, these kits were rushed out. I mean, they're really good kits. I mean, their sensitivity and specificity of the kits is really remarkable, considering how quickly they were made and tested and, then reviewed and then approved by the regulatory